So we're going to go ahead and get started here. Welcome everybody to our Research Ethics Week panel. We have a really exciting presentation and then followed by a panel discussion today. And so the topic of this panel is Synergizing Applied and Basic Science Innovators in the Research Ecosystem. Just want to give a quick kind of plug for our next seminar. It'll be Declan Schrader and he will be talking on March 25th and will be in VDL 280. This is a rescheduled presentation from prior. So we're really excited to hear Declan's talk. And so before we get started, I'm just going to give a brief introduction to our two speakers giving the seminar today, and then our third panelist who will be joining us after their presentation. So Dr. Alex Armstrong is a veterinary anatomic pathologist scientist and assistant professor in the Veterinary Clinical Sciences Department in the University of Minnesota College of Veterinary Medicine. She works with animal models of human orthopedic disorders to better understand their pathophysiology and to help develop better diagnostics and treatments, particularly related to pediatric conditions. And then we have Dr. Susan Novotny. She's a senior clinical scientist at Gillette Children's and an assistant professor in the College of Family Medicine and Community Health. Her research is focused on congenital and acquired pediatric orthopedic conditions, and she seeks to better understand the etiology of these conditions and identify better treatments for patients and their families. And then joining us later will be Dr. Meggan Craft. She's an infectious disease ecologist and professor at the University of Minnesota in the Department of Ecology, Evolution, and Behavior in the College of Biological Sciences. She leads a lab aiming to understand infectious disease dynamics in animal and human populations using a combination of fieldwork and modeling techniques. And so with that, I will go ahead and turn the floor over to our speakers, and I believe Alex is going to be speaking first.
Okay. Can you hear me okay in the back? Okay. Hi, everybody. Thanks for joining us today. I'm excited to just share a little bit about my research team and how it's going to work together to have a high standard of ethics in research. So this is the group I'm talking about when I talk about our team. And so we have 3 members within the College of Veterinary Medicine. We're all in the veterinary clinical sciences. And then we work with Gillette Children, so Dr. Novotny will talk a little bit about their role. But between the 5 of us, we have kind of a diverse background and training, which I think that gives us a real strength ethically as I think about our research. 4 of us have PhD training, 1 of us is an MD, 2 of us are DVMs by training, and we're all researchers, scientists. So we've had a lot of different perspectives that we're able to take on the work that we do. It's been really a pleasure to work with us in a group to consider these issues. To talk a little bit more specifically about our group in the College of Veterinary Medicine, many of you may know Dr. Casey Johnson. He's an MRI physicist. So in our projects, his role is mostly involved in non-invasive assessment, so MRI imaging. And that may happen in vivo, so in life with our generally pigs that we work with, our animal models. And then he also helps in the translation of those techniques from the animals that we use here to the children at Gillette. We also work with Dr. Tope. He's a large animal surgeon. So for our preclinical work, we're often doing surgeries on animals, piglets, to induce or model a disease, and then we may also be treating that. So Dr. Tote's role, he's a DVM like myself, so we both kind of bring that perspective to our work. And then he's thinking a lot about keeping the animals comfortable and how to have a really humane experience in life. And then I'm an anatomic pathologist, so maybe a little bit unusual person to be talking about the ethics because I usually don't see the animals during their life. I usually see them post-mortem. But to me, I feel like strongly motivated to make sure that our animals are used and used well. And I do all the post-mortem assessments. I may do a gross necropsy. I may do microscopic assessment. And then I'm really passionate about adding some more molecular assessment. So that's tests where we can actually go farther with the samples we're already taking to make sure we're getting as much as possible out of them. I feel like that's kind of one of my ethical burdens and ways that I can contribute to the team. So just a little bit about our shared work. So we mostly do NIH-funded studies. So they're considered kind of high translational value. They hopefully will have some benefit for our animal population, but the focus is also very strongly on how can we benefit people, and especially children who have the diseases that we study. We look at things like pathogenesis of disease, risk factors, why some animals and therefore people might have disease that gets worse over time rather than better if we can identify some of those risk factors. We do the imaging features, so how we could be more precise in diagnosing things and following disease through its course with imaging. And then the goal overall, right, is to find novel treatments and to treat children with these diseases better based on what we find in our animals. So because we're thinking specifically about children, we often use quite young pigs for our studies. They tend to be about 6 weeks old and older. So there is a little bit of an additional ethical burden for us of using these young animals, especially working in a College of Veterinary Medicine. All of us here are very passionate about animals and making sure they're well cared for and have good lives. So to use young animals brings, I think, a little additional burden, and we have to feel really well justified in using those animals in these studies. Most of our studies kind of start, as I mentioned, with a surgery to induce an injury or disease. We may do an additional surgery to moderate or treat that injury. We do advanced imaging or MRI, and then we'll do euthanasia and collection of the samples. Some of those samples are very focused in the area that we're looking at, especially because we do orthopedic studies. So there are opportunities with that to collect samples for other things. So for me, as a pathologist, I think a lot about kind of the end use of the tissue, but I am also thinking really deeply about the models that we use. So I think a big part of my burden ethically is making sure we pick a model that's really well justified, where there isn't a good substitute for it. So there are a lot of reasons that we use pigs. Pigs are a really strong model for children. They have similar size and anatomy. Their physiology and genetics are closer than a lot of the other animal models used. I feel like small animals like mice don't have as much similarity. And there are a lot of advantages to them. They also provide a lot of tissue for downstream analyses. And they're relatively easy to translate to our human studies, especially with the emphasis we put on the MRI, the imaging part of that. So to me, ethics isn't just about using fewer animals, but using the right animal for the job. So that's a big part of how that manifests in our group. And I just wanted to touch briefly on kind of the 3 R's, because that's what we think about in preclinical work, especially, is reduction of animals used, replacement of animals used, and the refinement of the animals that we use, and share for you in our team what this looks like. So obviously we use pigs in part because there isn't a great alternative. We can't access tissue from the children who have these diseases. It's just not available. It's not ethical. We don't have another good substitute for kind of the complete system and the bone and the cartilage and the growing aspect of it that is really important for the children we work with. But we have to know the literature, right? There are advances all the time in what's available as far as technology. So a big part of our job is staying up on the literature and being willing to try new things. I recently put in a grant exploring using the culture models, actually collecting some of the growth cartilage and growing it in culture, so outside the animal. And that would be something that we could then test treatments on in a dish before they would go into animal model studies, and hopefully help us reduce the number of animals we use that way. And then we also use cadaver samples. So a big part of our work ethically is planning our surgeries. Some of the surgeries Dr. Todt does have never really been done before. He has to be very creative in how we create these lesions to model the human disease. So we'll often use cadaver samples first to test out our surgical approach. And we can get the samples from the body donation program here at the CVM. It's great to have that. We can also get samples from the diagnostic lab. So if you're not familiar, they have a protocol where you can actually request samples for research purposes from cases submitted for diagnostic workup. And if it's something that they didn't need, they will give you those samples. So we're able to do that, especially for things like limbs. If we want to practice drilling through a growth plate in the limb that may not be involved in the necropsy, they're happy to give that to us to do that kind of pre-work. The other thing I'm really passionate about with the 3 R's is reduction. So a big part of this is doing our homework, working with a statistician. We have Dr. Aaron Rendell here in CVM, and he's a great resource, as well as the other statisticians at the EU. But making sure that we have the right number of animals in our study, and it's not just about having as few of the animals as possible, but also having a well-powered study. So if we don't have enough animals, we can draw real conclusions from our work. So both working with experts on the right number of animals to have a balanced study is really important. And then for me as a pathologist, finding ways to do other things with our cadavers. We often do a fairly limited necropsy for the studies that I'm on. So I've been really passionate about finding other uses for those animals after we collect what we need. And we've been able to work with the teaching team here to make sure those animals are used. They can be used in wet labs. They can be used with clubs. The pathology club has used them for necropsy demonstrations. So that's something that I really am excited to have those opportunities here to make sure they're well used. And then refinement, being embedded in a College of Veterinary Medicine, we have lots of DVMs here who are doing research or involved in the research process. The CIC and RAR, and then Dr. Tote and myself having veterinary training. I think we're especially attuned to making sure that we keep a high ethical standard in our large animal studies. So those are kind of the main things that I think about here with our work, but I'm going to hand it over to Dr. Novotny to talk about the human side.
All right, can those in the back hear me okay? All right, thank you very much for coming here today. Thank you to those who are on Zoom. I'm going to talk a little bit more about the clinical side of our research relationship for the next few minutes. And I want to talk about 3 different topics as we talk about ethics in terms of breakthrough research and building a team. This is largely for the trainees in the room. How many do I have here today? If I raise a hand. Just a few. As well as, so these topics are probably not new to many of you who are established researchers. But the third topic I want to talk on today is an emerging concept in medical research about fostering engagement, which might be a really interesting topic to facilitate conversations as we go on today. So what do I mean by breakthrough research? When we talk about this, do you really have what you need? And when we think about our traditional research team structure, which is different from what Alex just described, it's thinking about that individual lab. You have a PI who directs the vision, what experiments are going to be run. They've developed a team of trainees and staff who run those experiments and then generate data for the next proposal. When we think about that siloed sort of structure, there's some ethical considerations we need to keep in mind if we want to retain that structure. Sometimes it works wonderfully, other times it doesn't. When we do research in that way, we sometimes have a narrower scope of expertise. The limits of the expertise in that room are based on the team that you've built as well as yourself as the PI. And it puts us at risk for creating blind spots. We don't know what we don't know, and we might run experiments that lead to false conclusions or things that aren't repeatable or things that can't be translated into the clinic because they failed to consider something. That can lead to a prolonged time from bench to bedside. It can really be prolonged in terms of running that breakthrough research. And an additional core problem that we have in medical research is that complex disease requires various viewpoints of expertise. No single discipline alone can do this by themselves. We're going to fail every single time. And that's why sometimes implementation of research oftentimes takes 10 to 17 years. You'll see different quotes based on whether it's drug-related research or orthopedic-related research. So an example of this is what we study, like Hep-Perthes disease, and you don't need to know anything about this disease besides it impacts the hip and it's at risk for deformity. You can clearly see that. You should be an orthopedic surgeon and see that hip doesn't look normal. And for the last 100 years since we've discovered this disease, it's primarily been led by orthopedic surgeons and their reliance on x-rays. That is their tool of choice. That's what they use consistently. And so the prognostication of this disease, the treatment of this disease, and its efficacy of those treatments is based on x-rays. It may not be the right tool for the job. So when we started treating patients at Gillette with Perthes disease, they would be dropped off by their parents and they would stay in the hospital for 1 to 2 years and they'd be picked up at a later date. They'd be in bed for the most part or in bracing to promote healing of that hip. It's arguable that in today's times we haven't advanced this treatment one bit. This is our current patients looking the same as our patients 100 years ago. And one of the biggest ethical considerations we have is, are we helping children become children and healing them so their hip is a nice round shape, or are we actually creating patients? It's arguable that we're creating patients. So perthes is a complex medical condition. The more we learn, the more we realize how complex it is. And while I'm not going to go over all the details in this slide, is orthopedic surgeons alone don't have the knowledge base to address any of these other challenges that are considered in this slide that are important to this individual. So we really need that various expertise from large groups of individuals. So that leads me to how to build a team. And what do I mean by that? To conduct breakthrough research, you need to be able to reach beyond your single lab. You need to think about what it is that your blind spots are. You need to have a good, strong ego and a good sense of yourself to be able to say, what am I strong in, and where are all the areas where I'm not strong? So you can identify who those missing experts are, those missing techniques you need to do your research to get the right results. It might be, even if you have the right tool or instrumentation, does your lab have the capacity to take on that work? What do you really need to be a compelling candidate for NIH funding? So that leads to the evolution of an expert-focused or research team, or like a cross-disciplinary research team. And again, many of the seasoned researchers in the room probably have this well established, that there's different experiments you need, different talent pools that you need, and you found those key collaborators to be your collaborators. So you're basically identifying where are your gaps and putting a face to that name. So that together, you come together as a core group of individuals and solve this problem collectively, bringing your expertise to the table. But even this has gaps. Sometimes the more we work together, the more we know what each other's going to say, and we maybe are still missing something further. So for those who are in your trainee stage, as you travel to conferences, I encourage you to meet every single person who comes to your poster, get their name, send an email to them, because down the road, they might be someone you bounce an idea off of later down the road. They might be your national or your international colleagues who practice something differently than you do, or have different constraints on their government, how they can do research. And so it might open up an opportunity to think differently if you can collaborate with them. These are just an example of people we talk to almost weekly when we talk about clinical-based research and for these. So the last thing I want to bring up is fostering engagement. This is, again, a newer concept in medical research, but an incredibly important one. So in the early 1990s, there was this movement called Nothing About Us Without Us. And this evolved into what people know today as the Americans with Disability Act. And so the slogan of this campaign was, no policy, program, or law affecting people with disabilities should be created without their full and direct participation. This is integrating into medical research, and it's arguable that this statement should be revised to include medical research. If we're going to study a population, we probably should include them as part of the conversation. And so at Gillette, we practice our research patterns where we have what are called lived experience partners. Our patients are part of now the care team. We now have little Leah joining us as part of our care team, and in her area of expertise, her hammer in this game is her life. She's going to be telling us how to integrate this into our research. So lived experience partners are truly partners. They are co-investigators, and you must treat them that way. They are not participants in your study. It's a very clear distinction. You can't just do that. That's tokenism. We really need to involve them and listen to them and have them part of the process. There's many advantages of having them as part of this conversation, and some of the key things that they do is they provide perspective on their priorities. Your priorities may not be their priorities. You might be missing the mark in terms of how you're directing your research program to change a disease when it's not important to them. That won't translate into the clinic. They also stress test your assumptions. You may believe something for a very long time until a group of patients tell you, no, that's not right at all. That's not what we're experiencing. So it challenges you. It also identifies points of friction, especially in clinical research, where you might say, we would like you to come in for 6 study visits over a course of 2 weeks. And they may say, yeah, that's not feasible. I'm not going to participate. So they help you just identify your silly weaknesses like that, but some are more profound, that help you see things that no one on your research team had previously identified. So ethically, when we place this context of involving these lived experience partners into our research team, it's truly risk mitigation. It's truly risk mitigation, and it drives ethical, patient, and family-centered research in a very kind way. So to give you an example of working with our lived experience partners in the realm of perthes, I've created a group of them over time, both adults with perthes, parents of children with perthes, and then we're trying to include the patients themselves. And ironically, when we talk to them, they consistently say, Susan, I really do not care about the shape of my hip, which is what we've been concerned about for the last 100 years. They're like, I really want to talk about pain. Can we talk about pain? I've been sitting in these meetings with experts from around the world for the last 10 years, and no one has ever really talked about pain. We're missing the mark. We're missing the mark. And so that led to me recognizing that I have a blind spot. And so now what I'm actively doing is trying to find pain experts around the world who not only look at animal models in terms of the neuroendocrine aspect of those that are looking at nerves and blood vessels and bone, but also looking at who can help me from a perspective of pediatric chronic pain rather, as well as adult chronic pain. So we can start to measure this, address it in a meaningful way. So why should you care about this? Why should I convince you to even consider this? Funding agencies are beginning to integrate this into the competitive nature of grant submissions. PCORI, which is one of the nation's grant agencies, requires it. What a proposal for a cellular-based scientist versus me as a clinically based scientist might look incredibly different, but they require it. You have to have some program, and you have to demonstrate how they're going to be in there and provide a meaningful contribution. In terms of the NIH, the first institute that's making it a priority, so meaning like you have a bonus on your application if you've included it, is the neurological stroke, or disorders of stroke. Neurological disorders and stroke, excuse me. So that's an evolving concept that will hopefully be integrated into more of the NIH's institutes over time. And if you can integrate this sooner, it takes a long time to do. It's not something you can just easily integrate in like a new PCR test or a new genetic assessment. So hopefully today I've walked you through to get breakthrough research. I'm encouraging you to look for your blind spots, to build that team, and to consider fostering engagement so you don't answer the wrong questions in that breakthrough research.
All right. Thank you, Alex and Susan. That was really, really interesting. And I'm going to welcome Meggan to join our panel now. And so I guess I'll start by just seeing if there's any immediate questions in the audience. Otherwise, I have some pre-written questions, but I don't want to skip over. Okay, seeing no immediate questions, I'll go ahead and start. So for our panelists, the first question is, what ethical concerns have been the most pressing in your career, and what tools have you relied on to navigate them?
So I come from a very different perspective than my colleagues up here, and we are working with free-ranging wildlife. And so we're going to kind of switch gears here and talk about things that can't consent and don't have opinions about what you do and things like that. So I run a study on SARS-CoV-2, the causative agent of COVID-19 in white-tailed deer. We had been funded to ask the question, could white-tailed deer become a reservoir of SARS-CoV-2? And I have been funded by USDA, NSF, and LCCMR. So these, again, are complementary and different funding mechanisms than what we've heard about earlier. NSF might fund more theoretical work or basic science. LCCMR needs to be relevant to the wildlife in the state of Minnesota. And USDA got some SARS-CoV-2 American Rescue Plan money to deploy for studying SARS-CoV-2 spillover into wildlife. So that's how we got that funding. My research team is also interdisciplinary and varied and from different, I guess, layers of institutions. Let me explain what I'm saying by that. We have a national team where this study has been replicated in 7 other states. So we have a really great cross-site study design. And then at the local level here at the University of Minnesota, we have PIs from 3 colleges. So I'm in the College of Biological Sciences. Tiffany Wolf, who's in the audience, is here at the College of Veterinary Medicine. And our 3rd PI is James Forrester at in the College of Food, Agriculture, and Natural Sciences. I think I said CFANS, right? So we have a movement ecologist, a disease ecologist, and a veterinary epidemiologist on the team as well. And then we do community engaged research, not necessarily with the deer per se, but the people that manage the deer. And so we work closely with Three Rivers Park District, the managers there, and then also the Shakopee Mdewakanton Sioux community as well, and how to manage their deer. So the study design we have been contracted by USDA to do is to radio collar 40 deer per site and do repeat sampling for SARS-CoV-2 in those deer populations. And currently we have 4 sites that we're working on, and working on a 5th one at the end of this month. So hopefully we'll increase our sample size. And currently we've had up to 120 radio collared deer in the Twin Cities metro area to do this study. Prelim results are that white-tailed deer probably cannot sustain SARS-CoV-2 transmission in the absence of human infection and reintroduction of disease into their populations. So maybe we don't need to worry about them becoming a reservoir for this disease, mutating the virus and transmitting it back to us, which was our initial concern. In terms of some of the ethics and decisions we do that are similar to what I heard earlier is anytime we have a hands-on a wild-caught alive deer, we maximize the amount of samples we can take from that individual. So while this is a SARS-CoV-2 funded study, we are taking all the biological samples we can and are leveraging those samples to study other diseases and other pathogens in this population. And we're also banking those samples to be able to study future and emerging diseases that might be in the deer population as well. So we're trying to be really judicious with this precious moment to handle a wild caught deer. And some of the other ethical concerns we have to think about, again, is like minimizing risk, because handling animals is an inherently risky undertaking. And so we're trying to make sure that we don't cause harm to our animals that we're using in our study as well. And so I think the question is, what's some of the major ethical issues you're thinking about? And so one of the ones we're thinking about is like, well, I mean, this is a real situation that came up to me, is our Department of Natural Resources, who I need to get permits for, again, some regulation there, said, what is an acceptable level of mortality for your study? Is it 5%? Is it 10%? Is it 2 deer per site? Is it 1 deer per site? And that was a really challenging question for us to figure out. And I should also mention that white-tailed deer in the metro area are considered like a nuisance species by these managers. Our ecosystems have been disrupted enough that there's no natural predators of deer besides cars, the managers say. And so managers are actively paying sharpshooters to depopulate the deer population in order to make sure the trees can grow in these ecosystems. So it's an interesting tension there of here you have something that's maybe considered a pest, and then us that we're trying to do research in the most humane and ethical way possible. And it is not our job to do population control for the managers as well. So that was an interesting conversation for us to try and figure out how to like set that threshold and kind of deal with, again, inherent risks in handling animals. I think I'll stop blabbing by myself for a while and hand it on over. Do you want to talk on that?
Sure. Great. I can go just to follow up on that. So I thought it was really interesting what you said about collecting as many samples as you possibly can. So I think for me, that's one of my biggest struggles with the research that we do as a pathologist, especially as the research climate has gotten more and more restrictive with funding. that as a pathologist, I would love to do a full necropsy on every animal on our study and collect all these samples, even if we don't have a plan for them right now. But as we get more restricted in like storage space and the costs to process all of these things and do it in a way that they actually will be useful down the road, it's not necessarily viable for our lab, at least at this point, to have like a giant tissue bank of all the tissues from every pig that we've ever had on a study, even though I think in an ideal world we would do that and they would be available for follow-up and spin-off studies. So I've been trying to find ways to collect more things and do spin-off studies from different sites and things like that. But that's a reason things like the body donation program and the teaching and labs and clubs like here at the college have been a really great resource, as it kind of takes away some of that guilt that we can't collect all the samples we would love to. We can't save them in a way that would be useful. So finding some other avenues has been really great. And I think a lot of that is the resources here. Yeah.
Do you want me to answer this? Okay, so coming from a clinical perspective, one of the most challenging things is we're working with children with rare diseases. There's only so many studies we can approach them for within a given year. There's actually regulation on how many studies they should participate in. A series of surveys is easy, but if you're actually collecting samples, there's only so many times you should touch that population. And so we have to really be strategic about what we want these individuals to participate in, so we don't use up all of our cards or we don't get them to fatigue over time. Because these are conditions or treatments that we're trying to determine the efficacy of. So we're going to be asking them questions for a very long period of time into their adulthood, because they do not do well as adults. We know that. We know that's going to be their course. And so we have to be very mindful of that. At the same time, their parents are so desperate to make their lives better. And so we have to balance that ethical consideration of consent in terms of what should we really get them to consent to? What are we really asking? And maybe we should be designing studies better up front so that we can bank some tissues for down the road and grab those samples at a time that's not bothersome to them, maybe while they're already sedated. Is it blood while they're already getting an IV put in for their surgeries? Like, are there things we can be doing strategically to minimize that risk and to get them to be lifelong participants that we can continue to follow up on?
Thank you. I'm going to follow up with the question, and I'll add the caveat with, aside from collecting extra samples where we can, because I feel like that has been a general theme. But what are ways that you have enhanced what we learned from study subjects beyond the immediate study to maximize the science generated?
Maximize it. Okay, so I think, we've really been taking the approach of like reach out to any other collaborators running studies and see if they want to partner with us. And so we just got some results on orbiviruses, so blue tongue and episodic hemorrhagic disease. So that lab's super excited that we collected some deer samples and we're partnering with them. So I think that's been a way to really maximize study, and they've got different pots of funding as well. So maybe that's like diversifying the funding portfolio to try and keep the project going, as well as really tapping into a network of other researchers. So we've got that, spin-off projects, 10 spin-off projects off this. That's really helping us stay afloat as well. And I guess I was also thinking, we also try and do the minimal amount of handling as well. And so also work with a statistician. It's with the kind of observational fieldwork type of stuff, we can't really do some of the great stats with like controls and power analysis and stuff like that. So general rule of thumb is you want to get 40 collars out per area you want to say something about, and then 10 of those collars are going to fail and not work, so you're going to be left with 30. And so we are really trying to minimize those amount of collars and handling needed out there. And what we can then do is, so at Elm Creek, let's say we got 40 collars out, yay, that was great, but maybe there's 400 deer in that park. So we use mathematical modeling to scale up what we learned about those individuals, to the population level and start to simulate disease dynamics across this bigger population that we did not get to sample. And so that's where we can use some like quantitative tools to help really like leverage the empirical data that we get from the animals we handled. I guess I'm also going to say that moving forward, we are looking at using more non-invasive sampling techniques for the next phase of this project. So we'll be collecting deer feces and doing population genetics on the wild deer, which will be then minimizing the handling on those deer. And again, we'll be leveraging the data we got from the GPS collars because we're starting to think we're getting a good idea about how deer move in different types of urban landscapes from the current study. So leverage that data, then move on and ask new questions. And our new questions are ones that, that we can sample non-invasively. So that will be a big help.
I'm just going to repeat the question for the people on Zoom the best I understood it. So the question is, when it comes to your biobank samples, currently, are you just testing them for specific pathogen, and do you have plans to use them kind of down the line for molecular changes to apply those translationally?
Yes, so we are taking, let's see, we've got an ear punch, so it's a tissue sample. We've got saliva swabs, we've got nasal swabs, we've got feces sometimes. We've got blood, and then whole blood plasma serum. And currently we are testing for CWD, SARS-CoV-2, Powassan virus with Matt Eliota, EHD, blue tongue, and Lyme, anaplasma, maybe another tick-borne pathogen I forgot about. And then we're taking one of those swabs and doing, I'm calling it viral discovery on it. So just sequencing for new pathogens. And I'm sorry, I forgot the name of what's been found, but it's a virus that's not normally found in deer. So we were interested in trying to find a virus that was not even a virus, a bug that was common and apathogenic that we could trace transmission through the population using this common apathogenic thing. And we think we found it, and I forgot the name of it, so I'm sorry about that. But so kind of doing some viral discovery there as well. And in terms of like the molecular markers about detecting infection, I'm not super familiar with those techniques or what would apply there, but if you can use whole blood or sera or something like that, I'm sure we could collaborate with somebody to do what you're talking about. Yeah.
So again, I'll repeat just for the people on Zoom. So it seems like the core of the question is that as you ask your own question, as you're planning your study, other questions come up, like with the DNR saying that deer are considered a nuisance and they're overpopulated. So how do you determine from your own ethical viewpoint how far to chase some of those and how many tangents you can go off on, because there's always more questions to ask and answer. Did I capture that appropriately, Kathleen? Okay.
I think that would be a good application if we wanted to look at their nutritional status. For example, we hire out the capture of these deer to a helicopter capture company, and they catch them by net gunning or tranquilizing the deer. And at one point they were saying, gosh, these deer, they are acting different than the deer in Colorado or Utah or something. And so they actually have a hypothesis that these urban deer are kind of fat and weak because they don't get chased by predators and because they eat hostas. So yes, the hypothesis is already there that they might be deficient in some nutrients. So yeah, there's potentially that idea there that there could be some different nutritional characteristics of these more urban deer that are not living in rural areas.
So the question is more now along the lines of the deer population not being a good reservoir for SARS-CoV-2. And so what are your thoughts on that?
Yes. Okay, perfect. So yeah, when we first started this study, deer in some parts of the US had ridiculous rates of infection, like 60 to 70% of deer would have been actively shedding SARS-CoV-2 virus. And the study showed that these deer originally got it from humans. You can sequence the virus and you can see that it spill over from humans to deer. But then these deer could maintain the virus within their populations for, I don't know, 6 to 9 months after the spillover event. And so that was what was leading us to think, well, what if they can keep maintaining it in their population indefinitely and then that virus changes, and what if it then spills back to us? That'd be really bad. And it looks like these. So we haven't seen that high level of infection since that initial wave in the deer. The deer in the metro area, since we've been studying them, maybe have 30% prevalence. The males are more exposed than the females. Older more than the young. And we are thinking that these animals, that these males, they range farther. And we also found out that the deer that hang out in the neighborhoods are more exposed to SARS-CoV-2 than the deer that hang out like in the center of Elm Creek Park Reserve. And so it does seem that the deer associated with human habitation in the Twin Cities are more exposed to SARS-CoV-2. And we found that trend across the nation as well in this replicated study. We've also done some modeling studies where we now see how these deer are contacting each other through the GPS collars, and we are simulating the spread of SARS-CoV-2 through a computer population of deer that act like the ones we collared. And we're seeing that if you drop in infection in that deer population, it basically burns out because the deer aren't contacting each other enough during the short infectious period of SARS-CoV-2. And so it's kind of burning out. And so we're thinking that those big waves were either caused by a different virus host, like maybe that was just more, that alpha strain was more transmissible in the deer, or that it really just needs, there was a lot of human to deer repeated spillover then. And so right now we think it's human to deer spillover that's keeping these low levels going along. And our next part of the modeling exercise is to figure out like, well, how many spillovers do you need to replicate the seroprevalences that we see in the deer population? So that's gonna be where we go next. So take-home message might be maybe people should be reducing their contacts with deer. And we have a social science project that goes along with this where we're asking people how they interact with deer. So that's another collaborative cross-disciplinary thing we're doing. And it turns out a lot more people than I thought hand feed deer. So, or maybe put out compost that has your virus on it, or maybe there's some of these indirect transmission routes as well. And so maybe that's what's been fueling some of these outbreaks.
Thank you for those questions and answers. I'm going to pivot a little bit. So hopefully for Susan and Alex, and also Meggan, I would love to hear your opinions on this too. But so we all strive to curb unintended consequences, but zero risk is rarely a reality. And so how do you define an acceptable level of harm or risk, and who should make those decisions?
When we think about our patients, we're really thinking about what's going to cause them pain or additional pain or put them at risk for worsening their condition. And so we don't really want to put them into that situation unless there's a way we can control that. And an example of that is we collect biopsies for Alex's OCD work, where we're going to collect a biopsy before this child has surgery in the path line where a screw would be placed anyway. So that tissue is already going to be obliterated. And so it's easy for families to say, yes, you can take a biopsy of my child's knee or ankle or elbow because that tissue is already going to be destroyed in the pathway to fix it. And so that I find acceptable. But just to take a biopsy for biopsy purposes, I got to really think about that. You got to really convince me that that's the right thing to do. And we have to make it worthwhile for the family and make sure that that's going to be safe. And if it's not safe or it puts them at risk, what is our plan to then get them back to being healthy again?
The question also included like who should make those decisions as well. So maybe I'll follow up a little bit on that. I would actually say I feel the same way about you about the acceptable level of risk when you're taking samples. You know, let's take stuff you're already going to do. So yeah, the ear punch, the tissue sample we take is actually the hole for the ear tag too. So like a dual purpose thing there as well. So in terms of like who should make those decisions, I was actually thinking about the amount of like different stakeholders or regulations I have with my work as well. And so I'm funded by USDA. So that's a federal organization. And they're the ones that told me catch 40 deer per patch, sample them multiple times. And here's the sampling protocol. So there's some, I guess, some federal oversight there. And then I have a Department of Natural Resources permit. That's about 5 pages about how I need to handle that animal and what samples, what I do with the different samples and who I notify if things go wrong and stuff like that. So that's 5 pages there. And then I've got Aya Cook in there for the ethical and welfare of the animal. And then I guess we have 3 PIs with diverse backgrounds as well, also making decisions about how to, what samples to take and how many animals to handle as well. So yeah, I feel like there's a broad team of people trying to all do the ethically correct thing.
Oh, yeah. I mean, for me, it's very much a team thing, too, in that I'm the pathologist. So when the animal's already been euthanized, my part of the job, there's less risk, I would say. Pretty easy to say there are acceptable levels of risk when we do things that we've done many times before and not going out on a limb very often. But for me, especially being a PI of a study where I'm not a surgeon, but I'm asking someone to do a surgery, I rely very heavily on people like Dr. Tote and CIC, Caitlin, all of my partners in the process to tell me, like, is this possible? What do you need to make sure you can do it in a safe and an ethical way and minimize the risk as much as possible? And do you feel comfortable even doing it? Because if you don't, then I'm obviously not going to submit a grant saying we're going to do it. So doing your homework on the front end so that you propose something that's feasible and safe, at least to the extent you're capable of controlling it, is really important.
Great, thank you. So this was an audience question that actually got submitted in advance, and I'm going to add a second question on top of it too. And so the question is, in your view, how important is private philanthropy, so donations from individuals, in the context of the overall medical research funding landscape as compared to traditional sources of research revenue, so grants and industry sponsors? And then my additional question on top of that is also, do you find that there are additional pressures from those individual donors to produce some sort of product or outcome compared to the private funding, and how do you navigate those if those exist?
When we're working with clinical populations, these patients are ultimately going to become hopefully our donors. If we're doing a good job, we're almost dating a donor from the day they're diagnosed. Their families are donating to us. Some of their grandparents have passed and then donated money to us to make their grandchildren have a better life. And so from a clinical perspective, philanthropy is part of my job. It's just as heavily thought about, and I prepare for that as much, those interactions and those lunches and those meetings, as much as I prepare a grant application. And so for me, that's a parallel line. I'm not only relying on NIH because I don't know that that's going to be a fruitful source for rare disease in pediatrics forever. And so it's much easier to work with donors and philanthropy. So we have a philanthropy board that is separate from our board of directors at the hospital. And they're in charge of gathering our ideas, what we want to study, and actually finding those donors for us. And so we're constantly giving them ideas of where's our research going in the next 5 years. And they will often, if we do get grant funding, find a donor that'll match it. And so we're trying to capitalize on those relationships. And so relationship building is incredibly strong from the clinical side and will probably be grown at a faster rate than our federal funding. And so we're becoming really reliant on that. And so I probably have a greater comfort in that kind of a realm than others, and I'm interested to hear you guys' perspectives.
And I guess for me, I'm pretty far usually from those decisions of funding, and I work mostly with NIH funding, but for studies I'm doing where there may be, I guess, an expectation related to the funding of what the outcome is, I think the biggest concern for me would be communicating appropriately kind of what the expectations are for outcome. I use these techniques like spatial transcriptomics, which is a novel emerging technique. It's very new as a platform, and it can be difficult to use, especially in our samples. Like some of the analyses we're doing have never really been done before in the tissues that we're collecting. So something that I strive to do and that I could do better in, I think, is giving realistic expectations to the sponsors that we work with, that like this may or may not produce anything relevant clinically, and that's a risk you take. We are open in like our IRB approved studies that this will not benefit the patient now that we're getting a sample from. And they only consent because they know it may benefit somebody down the road, but there's no guarantee of that. So being upfront about that is really important.
We've had good luck with private donors in the past, especially when I was working with more charismatic megafauna. So with the Florida Panthers and African lions and stuff, I could get more private donations for that work. The white-tailed deer is a hard sell right now. So, so if y'all have any help, I know. But yeah, College of Biological Sciences and University of Minnesota, like we're not really ramped up to look for private donor money like that. But I think it would be very helpful for us to, again, to keep diversifying the places we can go for funding for this. The white-tailed deer, like, yeah, it's not a conservation issue, so you don't go to conservation. It's kind of a zoonotic thing, and it's also maybe a public health thing in terms of car strikes and human health that way. I mean, I don't know, maybe it's a conservation issue when you think about chronic wasting disease coming through the Twin Cities and us trying to figure out how to like understand deer movement and behavior and connectivity to see how a pathogen like CWD or a future emerging disease might spread. But again, it's unclear which of these like siloed funding agencies you go to for something like that as well.
So the question for those on Zoom was that the use of animals in research has come under really intense scrutiny in the, it has always been, but in the more recent past has come under more scrutiny with pushes to fully replace using things like AI, but it often feels like it's without input from the appropriate experts. And so how do you navigate that? And do you find yourself now in more of an advocacy role than prior?
That's a great question. And I guess in my limited experience so far, at least working with the NIH, part of my thought is to trust that there are still those people there who know, and whether it's my program officer or the people reviewing the grants, that they're hopefully still pulling in people who have that background and expertise to understand what gaps we're filling with animals, and that it's not something that's done nonchalantly to use them. Like if I never had to use a pig in a study again tomorrow, that would be terrific, right? That's all of our goal really long term. So I guess, and on that side of it, like in some ways trying to see the best of it, of like, it is good to be reminded that we need to keep advancing science and embracing alternatives. And if we can find ways to do more of that, while maintaining a high standard based on our training and our experience as scientists, I think that's good. But it is scary to hear blanket attacks on something, especially from bodies that don't understand, and it definitely harms the public perception of what we do. So I think communicating your science is as important as ever, of being able to tell people like, this is why we do what we do. I think we're also lucky to work in kind of the pediatric realm, where I think the people involved, whether it's patients or parents or caregivers, clinicians, like it feels like a really solid team of people who are like, we need to help these kids and we will do what we need to as far as the best way to approach that and not getting blindsided by public or political opinions. I think they have their eye on the prize to some degree.
I think one of the things you use the word advocate, but maybe we just need to advertise our science in more realms. It takes about 7 times for somebody to see something before they believe it, is a stat I've heard recently this week. So maybe we need to advertise ourselves more on social media or on things like Twitter, or where the community can see what we're doing and the value in it. And so maybe we do hold a role in broadcasting what it is we do well. Because in its absence, it's not being heard. And so maybe that's a role we can play in a way we can evolve the science and just make more of the community aware of the good we're doing, where maybe the only message they're getting is bad situations or mistakes.
So we'll switch gears a little bit, but kind of piggybacking off of Kevin's question. And this is technology such as artificial intelligence helping to solve our environmental and animal welfare challenges, or is it creating a new set of ethical dilemmas that we aren't prepared for?
I guess we use machine learning models to run our statistics for some of the research to figure out like what are the drivers of infection in these animals. So I looked it up last night. That technically counts as AI, but that's old school. We've been doing that for 10 years. And about anything more modern, I'm going to need to be quiet about because I'm not sure where this is going. And I'm pretty sure we need some human oversight in the AI field, but I can't speak more eloquently about that.
I guess my perspective on it is I don't fear AI very much as far as putting me out of a job, because all of us are the people who have to be there to rein it in and interpret what it's doing and decide if it's right or wrong. So I try to stay positive in thinking about it, as it's not removing a conversation, it's just changing a conversation, I guess. But yeah, I think it's definitely doing both things. As far as directly in our work, I'm personally not using it as far as any of like really my science, but I'm hoping there will be tools that come from it that can make us more efficient, especially when higher education and academia, we're underfunded as always, and we have limited time, limited people. If there are ways it can help from a resource standpoint, I think it probably already is more than I can appreciate. But definitely there are many ethical dilemmas, I think, that we'll have to be prepared for.
From the human standpoint, there's some decisions that we need to make and have some guidance on and how we utilize these things. We don't know what we're feeding the AI when we're doing things. And so we have a lot of collaborators across the nation and across the world who are starting to feed their MRI, for example, into AI and have it read it automatically and do those sorts of things. Can it do measurements that otherwise take an orthopedic surgeon half an hour to measure? I think we need to be incredibly critical and thoughtful about those kind of processes, because the back end of those MRIs and X-rays and whatnot might have information about those children in terms of actually who they are. And so we have to have a little bit more guidance before my team is going to jump into those sorts of things, that you can guarantee me that MRI you're putting into that software is actually truly blinded. Is there a way you can guarantee that they can't tie this back to this child and use it against them in a way that we don't fully understand 10 years down the road, 2 years down the road, 30 years down the road? The ethics committee here at the university is always about not releasing the identity of that patient. And that's one of my number one things that I'm so concerned on when we talk about AI and using it for measurement schemes, primarily of images.
Are there any questions in the audience? I don't want to just keep going to my paper if there are questions that people are waiting for. Okay. So in kind of speaking about the ethics review and the IRB here at the university, what, if anything, would you change to the current system in place for ethics oversight? So for on the human side, things like the IRB, and on the animal side, things like the IACUC, to make it more effective?
From the IRB side is what I primarily work with. I leave the IACUC stuff to Dr. Todt. It's becoming a process that's continually evolving. And the more it becomes evolved, the templates get longer, the protocols get longer, they make you think about more things, which is a really good thing. At the same time, as the standards improve or get higher, some studies that we've had going on for so many years, they make you re-put your stuff into a template that fits the current foundation versus now. There's a lot of redundancies in terms of wasted time that are administratively driven from my perspective that maybe are impeding our utilization of our brain time that otherwise could be put to good use. And so I think we really need to think about how often they make us do some of those changes, or can you be grandfathered in? Like, what are those things that maybe are administratively burdensome just to meet today's standard? If it's not going to change the risk I'm putting a child through, if it's not going to change the consent form, if nothing is really changing besides a document, is that necessary?
Yeah, we will have the same issue with the IACUC as well. So it's just, I don't know, if that took me a couple weeks to renew this year. And the form, I would say, is not designed for free-ranging wildlife either. So but I also can see the other side, like, is it worth making a form that has drop-down boxes for the free-ranging wildlife and fill it out? So it's a messy situation to try and figure out how to improve it. So I don't have a great solution on how it is, but investigator time, I would say, has increased.
All right. Another question, again, kind of shifting gears a tiny bit, but how do hours, the royal hours, or your own value judgments influence your decision making in the work that you do?
So I do have a child with a disability. And so when I think about my patients and their families, that's always in the forefront of my mind. I am a mom first and a scientist second. And so that really has changed my trajectory in terms of how my career has evolved over time. I wanted to be an Alex, and then my life kind of changed. And so in a very good and positive way, because I have a vision of how research is conducted from my benchside training, translating into the clinic. And so I have just a really unique perspective that's often leaned upon at Gillette and in my collaborators around the nation and around the world, that they'll ask me things as a mom, like, what do you think about this? And then how would you do this clinically? How would you then implement this in terms of an experiment? And so that really has weighed heavily into my lens by which I conduct research and how I think about research. And I'm less likely to participate in studies that aren't well thought out for my own child. And so I do say no to a lot of studies based on that, but I also can help add that to the conversation as we're thinking about how to design things in a really well-designed way that make people want to participate. And it's easy to participate, and it's not a burden to participate.
And I have the same kind of perspective as a veterinarian and a lover of animals. I think, especially in vet school, being a pathologist, people are kind of like, oh, you don't want to work with live animals. You don't want to be in the clinic. Like, you don't care as much about the animals. But I think from a personal perspective, I feel like I'm such an animal lover that it makes me kind of emotional to think about being involved in their pain and distress, especially clinically. So being able to be involved as a researcher, that was actually a big motivation for me to do a PhD, was to know how to do good research and research that's really well designed so that you're being really effective in minimizing the animal distress and the animal use as much as possible. So I think it's a really great perspective to have as a researcher, to be like, I really love animals. I would never kill a pig callously. Like, I take it very seriously when we do that, and I can be the one to like very firmly justify that and then make sure that we're keeping a standard that I think for myself is quite high.
Yeah, I think y'all's answers are really great, and I'm going to just say that'd be similar for wildlife as well. So those of us who go into wildlife, research is not for the money or the prestige or anything. So yeah, we do like trying to come up with some solutions to some of these tricky problems that would benefit, humans, animals, and the environment.
And I do think that's kind of a fun and unique thing for like being a researcher and being at a College of Veterinary Medicine or having veterinary medical training and going on to be a scientist, is there are many people out there who do preclinical work with animals who are not veterinarians by training, and they don't necessarily have that passion or just understanding of animal welfare that we do. So I think that's a really great thing about our community and a way that we can be leaders in what we do, especially moving into like the NIH and these areas where it's dominated by people with different backgrounds.
Can I add a second piece to that question? And do you find that your own personal values and your value judgments also influence the teams that you surround yourself with and how kind of choosy you are with collaborators at all?
Yes, definitely. I mean, like Susan said, kind of like choosing studies that you get involved with, that if you don't think that they have your same values and that values might be expressed through the protocols they write or the studies they design, it makes it much easier to say no, which can otherwise be really hard. So yes, definitely.
Yeah, yes, definitely. I mean, it's been fun getting older and more senior to be able to, I guess, have the agency to not have to say yes to everything and to be able to opt into studies with kind, no drama, smart people to work with. And that's been really fun. And also to put teams together of kind, no drama, smart people to work with. It's been really rewarding.
And these are long-term relationships you're building. You're going to spend more time with these individuals than you do with your family. And so if you don't, you got to choose people that you really enjoy working with, who really value you, who listen to you, who take you seriously. And it just truly is a friendship after a while. And so you really want to choose those people carefully. And especially once you start to choose graduate students, how do you vet those students to say, are they going to fit into the lab culture? Are they going to have that same kind of value system? That's really difficult and something I think we have a lot of conversations about, and how do you do that well? Because it can be when you have a collaborator who's not a great collaborator, it's a really long 5 years or however long that grant is, and sometimes longer because things don't get done on time. And so you're shaking your head, yes. And so you really just want to be really smart with those relationships and build the strong ones. And that means you have to be a good person to the rest of your team, always.
Great. I think in the interest of time, if anyone has one last question from the audience, we can take that. Otherwise, we'll thank our panelists and thank everyone for joining. All right, great. Thank you.